A big welcome to our viewers out there from the Wild Genomics Lab at West Virginia University. We're a conservation genetics lab that is focused on studying genetic diversity of fish and wildlife populations. And this is the first in a video series that we're doing. So as you're watching the video and you come up with questions yourself, feel free to comment and let us know what questions you'd like to see us address in future video sessions. We're going to be highlighting all the different people in our lab. I'm the principal investigator. I'm Dr. Amy Welsh, and we're going to start off with just a few questions that we'd like to address about things that might be on your mind about conservation genetics, or that you might want to know more about in terms of conservation genetics. So the way I first got into conservation genetics was I started the study of lake sturgeon when I was in grad school. And honestly, when I started that project, I wasn't a fish person. I didn't think fish were going to grab me, so I was kind of hesitant about where this would go. But I'll tell you, if there's any fish that's going to grab you, it's lake sturgeon. They're a pretty unique fish. They have a lot of prehistoric traits. They've been around since the dinosaurs, and they look like they've been around since the dinosaurs, so they're pretty cool. It's always neat when you see one in the aquarium and people will be like, what is that fish? And then, I mean, it really stands out from the rest of the fish in the aquarium. So pretty unique species. And so I did my PhD work looking at lake sturgeon genetics in the Great Lakes, and after that I was hooked. It's still my favorite species. You can ask my students. I always use them as an example in class, and they're pretty unique because they present a unique conservation challenge in terms of they are long-lived. They can live past 100 years. They don't reach sexual maturity until about 25 years old. So it's a challenge in terms of when you do conservation actions on lake sturgeon, a lot of times you're not going to see the effects of those conservation actions until many decades later. So it's definitely a species that you're in there for the long haul with, and it's a challenge in that way. Definitely a big conservation concern because the populations have been depleted across the Great Lakes and in Hudson Bay. So it definitely is a conservation concern, and that has always been where my true passion is, conserving the species that need it most. And so lake sturgeon has been that species for me and continues to be a species that we focus on in our laboratory. So now that, since lake sturgeon, when I first started at WVU, my goal was to expand into, I had worked on other fish species, but I also wanted to expand into wildlife species. So the projects we have going on in our lab right now, we have a white-tailed deer project, we have projects on bobcats, we've expanded into birds where we had a Cooper's hawk project, and we've had king and clapper rails. We're dealing with disease where we're looking at ticks and disease epidemiology. We have people working on amphibians, fracking in the Louisiana water thrush. So lots of different taxa. We've really expanded the different species that we've worked on in collaboration with lots of different biologists and scientists. Common thread is conservation of our native species. So some of these species are of immediate conservation concern. We're concerned about their future. Other species are game species, but we want to manage so that they don't become a conservation issue in our state, and we can preserve that natural heritage that we have here locally. One of the big questions I'd like to address in the future is we are now moving into the age of genomics. So we hear a lot about genomics and looking at genome-wide variation. A lot has been done with that in terms of humans, in terms of medical research, agriculture. We're just starting to get into that with fish and wildlife species. And now we can look at variation across the entire genome. We can look at genes that actually code for particular traits. We can look at the effects of selection. Do we see adaptive differences between populations? We can now look at gene expression differences. You may have a gene for a particular trait, but then the environment can determine whether that gene gets turned on or turned off. What does that mean for management? So I'm really interested in looking at adaptive variation, looking at gene expression differences, and then tying that into what does that mean for management? How do we manage for those differences? If they have differences in gene expression, does that have implications on how they're listed under the Endangered Species Act? Does that have implications for how we define our management units? So things are going to really change from a policy standpoint and from a management standpoint as we start understanding more and more of those layers of variation in the organism. So that's one of the big questions. So we know a lot of people feel that conserving biodiversity is important. Lots of reasons out there for why we should conserve biodiversity. Genetic diversity is an element of that. Why do we want to conserve genetic diversity? It's something that is hard to see. A lot of times we can't see that genetic diversity when we're looking at populations, and that's where our studies come in. And the reason we want to conserve it is because that is the foundation that allows these populations to adapt to a changing environment. And we are in a changing environment. So if those populations and those species don't have the ability to adapt, then our planet's going to look pretty different in the next couple of years. So we look at that on a big scale in terms of what's the evolutionary potential of these populations. We can also look at it in terms of, are these populations genetically healthy? Are they inbred? Not a good idea to mate with your relatives. So if they're inbred, we could be seeing consequences from that, and we need to manage for that. We need to identify what our different populations are. Maybe conserve those differences, or maybe bring together populations that have now been separated due to habitat fragmentation. Genetics can help us get at those questions that may be difficult to get at in the field, with the ultimate goal of preserving the genetic diversity within our populations and the genetic diversity across species and even at the community and ecosystem level. I would definitely recommend getting lab experience. So it's a combination. What's unique about our field is you need the skills to be able to do the lab work. You need the math skills and the analytical ability to analyze the data, especially as we move into the area of genomics. A lot of that is becoming heavy in bioinformatics, so a lot of that involves less lab work, but more time at a computer analyzing data. So having computer skills, having laboratory skills. If you're an undergrad and you're thinking of pursuing a graduate degree that's focused on conservation genetics, volunteer in a lab, get that lab experience, take some computer classes so you can understand some computer coding, and have those skills to set you apart from other people in the field. Many of my students do their own field work, so getting that experience, don't ignore that component as well. Getting that field experience and understanding how to collect those samples in the field. Field work's not your thing, that's all right too, because a lot of our work involves collaboration with both federal and state agencies, where we work together with them to get more samples for our projects. But definitely getting that experience for your resume. We get a lot of top applicants for our graduate program, and those that have that lab experience, that research experience, really stand out from the rest of the students. Well, thanks everyone. Tune in next time for our next video, and definitely comment on this Facebook post and let us know what you want to hear about. Thanks.